All right, and let me find Kennedy, make you a
Nice.
co-host. There we go. All right, can everybody see my screen? Good, good. All right, welcome to week 2. Kennedy, you've been made host, so if you could keep an eye on if anybody else jumps in, that would be wonderful.
Yeah, sure.
Thank you.
Mhm.
So welcome to week 2 of Biotech Infusion. Hope you guys have been having a good week so far. Some people are on fall break, so thank you for joining us if it's your fall break right now. Yeah, anything but being in the classroom, right? And some of y'all are holding out till fall break, so we are right there with you. You'll make it. It's okay. You are strong. You will make it. I wanted to go ahead and talk about a couple things before our speaker joins us. And Kennedy, that will be Megan Eckerman. So if she comes on, just let her hang out in the waiting room for a few minutes.
Okay. Okay.
So first off, one of the things that we built this training around was a set of essential skills and competencies. I will get this into the Google Drive. Right now, the document I have is a little bit old, so I want to make sure I update it for you guys. But whenever we go through different job openings for entry-level positions, these are the things that we see most frequently. They're not all on every single one, but we've noticed that these pop up most frequently. And so we have tried to bring you labs that highlight some of these skills so that you can get students ready. We keep hearing how important soft skills are and how important these are. These are kind of like lab soft skills. So we did the glow germ aseptic technique lab to get them thinking about keeping themselves free of samples and keeping the samples free of them. So that's why we use all the PPE and proper hand washing and all of those things. We have calculations and measurement. Um, so you guys will be doing a little bit of calculation today and measuring out the rubbing alcohol. So you'll get a taste of how easy that is to do with your students. Reagent and media preparation, again, measuring and diluting your alcohol definitely falls in there. Lab sampling procedure and practicing. So, for example, using the micropipette, that falls under lab sampling procedure. Anytime they're working with small volumes, like any research lab or individual testing, like you might see at a hospital, they're going to be using micropipettes to take out samples. Equipment use and maintenance is number 5. So learning to use all the different equipment in your lab. And then when they get to a company, that company will train them on their specific equipment. But if they already have some background in using that item, that's definitely a bonus. That includes microscopes are probably the most common one that you guys have. Record keeping and keeping a lab notebook. That's something that students really have to get used to as they go into college as well. Um, Keeping every single detail about anything that you do. If you made an error in what you added as far as the amount of the reagent, that's something that needs to be noted in your lab notebook. So getting them to start that now is great. A lot of places have moved to digital, but not everybody. So it's important to learn to write it. Um, let's see, keeping inventory. So that's something you could have your students help with. Maybe keeping inventory of different lab supplies in your classroom. I think we had them doing that in my classroom when I was teaching. I would say this is what's supposed to be in the cabinet. Can you guys go check that? So just giving them some ownership of their space. Next is safety, and really that could be number one, number one through 10. Keeping themselves safe and then keeping the sample safe as well. And then the last one is just following directions. So when you look at job openings, that is listed as following standard operating procedures or SOPs. So you can have your students work on not only following them, but maybe creating their own so that other people can read them and follow them. I think I've done that or had students do that with like peanut butter sandwiches before. And they take it very literally if they don't know what to do with the bread and the jam. So all of those skills really help with employability and showing that they're ready to work in a job. And I've done a lot of scraping different companies that we work with. A lot of them have a high school diploma as their minimum requirement. So a lot of times your students could be going straight from high school to getting jobs in these companies, especially if they come in with skills. That gives them a leg up. So the next thing I wanted to talk about is the Equipment Depot. If you are not familiar with this, this is the other program that I run. So we have a warehouse in Tucker. We take in donated items. And you guys can come visit the Equipment Depot. Sometimes we take the Equipment Depot on the road. And the whole point is to get you guys stuff for your classroom so that you can do hands-on labs with your students. I know personally that you are spending too much money on things for your classroom. I don't know a teacher who can't say that. So we try to do what we can to knock down your list so that you can buy the more impactful things that we maybe don't have in stock. This QR code goes to my Calendly. And so that Calendly, you can look, I don't know how far out it goes, but it goes out for months. And it accesses my Outlook calendar and looks at my availability. And you're welcome to schedule anytime on there. That one is Monday through Friday. But if you need to come on the weekend, let me know and we can try to work that out. If you need things and you can't make it, let me know and we will try to work that out. I'll talk more about it later too. So here's the address. It's the one that's in my signature. We are only open by appointment, so please let us know before you show up because sometimes we're just not there. I was not there all day today, nor was Kennedy. But here are some of the examples of things that we have there, and it varies from week to week what's available. Oh, somebody, I see the chat's going now. Cool. Y'all have that under control. But we get in all kinds of PPE. Right now, we do not have gloves, but we have so many face masks, surgical masks. We have plastic gowns that you're welcome to use kind of as lab gowns, but it's the healthcare kind that like tie in the back. We have all kinds of consumables, whatever people are getting rid of. If you come, bring your micropipette and we'll make sure that your tips fit before you walk out the door. So you're not taking home things that you can't use. We get in all kinds of glassware, plasticware. We get healthcare supplies. They figured out that we will take blood collection tubes and needles, all kinds of random consumables. Like I had an intuitive surgical little robotic arm thing the other day. I think that one left finally. But really, it's like people are throwing out all kinds of things that were never used. They're still in their package. So we know that you guys can use those to deliver hands-on labs to your students and get some use out of them before they go in a landfill. We also get really big items sometimes as donations, but we don't have space for that. And so in that case, we will connect you directly with the company. You can find any information on that on our newsletter. So if you are not subscribed to our newsletter, you get an email about once a month. I will put that QR code on the next slide. I didn't want a double QR code. Um, but we put what's new in the depot in there, and then if there are opportunities to go pick things up, we will let you know. So sometimes companies are getting rid of giant freezers, for example, or biosafety cabinets, which is a lift, but if you get your facilities team involved, they can go pick it up for you. So there is the newsletter QR code. A lot of you already receive it. It comes straight from my email now and says Educator Connection newsletter. We will try to get one out probably next week. Call it the October one. We're almost to October. Oh, scary. Cool. I don't see anybody else holding their phone up to scan that, so I think I'm safe to move on. And yep, that was the end of PowerPointing for right now. And we will have our speaker in a moment, but I want to give you guys a couple minutes if you have any questions. And I'll check the chat and make sure nothing was from me. Yeah, and Cynthia has all the resources. So if you need anything, ask. She's happy to provide. All right, any questions? Good for now. Well, and you guys know how to find me and Kennedy. We're happy to happy to answer any questions you have at any point. And my cell phone number is in my signature as well. So if it's easier, feel free to shoot me a text. If you're in front of the kids and can't get to your email, that's fine too.
Yes. Can I say one thing, Megan?
Yeah,
So, hi teachers. My name is Kennedy Duma, and I wanted to ask you guys when you come in here, can you put your first name and last name as your name? I'm taking attendance, and so sometimes I don't know like who you guys are. Which, so yeah, just first name and last name. No, I'm talking about as your name,
your Zoom
as your
name.
Zoom name.
How do
Yes.
we change it? Because mine is, I think it was already set from previous meetings, but I don't know how to go. Is it, hold on,
I
let me
think there's
see.
a setting. If you can't, then please
You
just put your name in the chat. And I know
Mhm.
like Wayne said that he's there with his wife Carla.
Oh,
So,
wait, I see
um,
rename. Hold on.
yeah,
aha,
you can press the
Thank you.
three
All
little
right, it's
circles.
rename. All right,
Mhm.
thank you.
You're welcome. Thank you.
perfect. Yeah. Oh, and now Carla has joined separately. Cool. So y'all are good. All right. Um, any other questions? Thanks, Kennedy. Keeping up
You're
with
welcome.
everybody's attendance. Make sure we see you all.
Yes.
All right. And Megan, did you join?
Yes,
There you
I'm
are.
here.
Aha. Awesome. Yeah. All right, everybody. So our speaker is here to join us. So I would like to introduce Megan Eckerman, and I will let her tell you about herself. Would you like me to bring up your PowerPoint, or would you like to do it?
I can bring it up.
Yeah, you should be able to share.
Okay.
And you didn't have any videos that needed sound, I think. So
No, I didn't.
that's for the best.
Yes. Okay. Can you guys see my screen?
Yes.
Okay. Well, first of all, I want to thank Megan for inviting me here to speak with you guys. This is my first time doing this presentation, and from what I understand, what I really want to share with you is just my career progression. How did I get into the life sciences industry? And, what could this look like for future generations as they think about, where they want to take their career and what they want to do? Um, like I said, this is my first time with this presentation, so keep it fluid. If you guys have questions, I may not be able to see everybody, but just kind of cut me off. And so I want to make sure that you guys are getting value out of this presentation as I move along. Sound fair? Um, Okay, so first, what I wanted to do is just give you a little bit about me and as I kind of walk through my career journey. So first, I want to start just kind of with my early years because a lot of this stuff is, you guys are working with your students, understanding their background, where they're coming from, and little things matter, as you guys are, kind of working through, these career conversations with your students. So for me, I was in a family where there were 7 kids, a quite large family for this era, and I was the third oldest. So, kind of grew up fighting for resources, understanding when teamwork, needed to occur and really, how to work together. So that was my childhood. I'll bring more of that up as we kind of get into, my education. But in middle school, I was a good student, kind of, high B's, low A's, but I was heavily involved in athletics. So soccer and basketball were my two sports of choice. And that competitive edge is really kind of something that I've leaned into as I've moved my career forward. And then in high school, good student, got really involved again in more sports. And then, started to get more involved in clubs. And so I took leadership roles in the Key Club, and then I was student body secretary. So just a pretty involved child growing up. And I think, some of the skills that I looked at that I developed in these early years that have helped me, kind of in my career and as I've moved it forward is just the sports, very disciplined, took leadership roles early on, learning how to communicate as such. Yikes. From that perspective. So that was a little bit about me growing up. Now, as I started to explore options for college, growing up in a very large family, my parents kind of said, "Hey, how do you plan on paying for this?" I was like, "Whoa, wasn't expecting that." So, kind of through some exploration, decided to pursue an ROTC scholarship. So I did come from a military family. My dad went to the Naval Academy, had other West Point grads in my family as well. So it seemed like a little bit of a natural step for me. I wouldn't say I was gung-ho military, but thought it could be a good stepping stone for me. And then it, kind of provided me with the crucial resources I need. So I'm not sure how much you guys are familiar with ROTC scholarships, but on a whole, you do apply for a scholarship for a certain major. So I had actually wanted to be a physical therapist growing up. That was what I wanted to go to college for. That's what I wanted my degree to be in. They did not have a physical therapy scholarship. So I decided to apply for a nursing scholarship and thought that I would be able to leverage that and roll over into physical therapy, later on in my career, college career. So I entered Indiana University, was my undergraduate college. I had started with my nursing degree. After about a year in nursing, I was trying to move over to physical therapy, and the military said, no, you've got to stay in nursing, or you can go over to computer science. I said, okay, let me try computer science because I don't love nursing. Did that for a year. Now I found myself in my junior year moving over into physical therapy. And so I had 3 more years to go on that degree. And the military said, hey, we need you in the military in a year. So do what you can to wrap up your degree, or we'll pull you in as an enlisted and not an officer. I said, well, let's see what I can do. So I ended up with a kinesiology degree. Um, so having that life sciences, kind of degree under my belt has helped me throughout my career, and you'll learn a little bit more about that. And then I did end up getting my MBA when I was in the military. So that's kind of what it looks like from my college exploration. I do have to say the military was an unexpected stepping stone that has been such a blessing in my career. Employers, especially in the medical device and med tech space, really like to see military experience, especially as you're kind of moving up through leadership positions. So it's one thing I wasn't really anticipating being such a strong thing for my resume and my career path, but it was. When I went into the Air Force, I was in Space Command. So it was the Air Force, and there was a subsegment of space within that, not its own branch yet. And I went to school for a year for mechanical and electrical engineering to learn how to fly satellites. And then I had 6 months of on-the-job training, so about a year and a half. And that training and experience has really played through my medical device career because I have the aptitude to speak, with engineers and with clinicians from a very, engineering mindset, which has given me the ability to translate kind of marketing skills into other skills. I was told by Megan that you guys would have questions about, kind of salary ranges for different positions as I went through. So keep in mind, I am several years out of my career, but as I was entering the military, I was only making $20,000 to $30,000 a year, but they paid, for my living. So that was kind of disposable income there. And then as I moved up into my fourth year, making about 40,000 with my living expenses. So not a horrible career for, early 20s. I'm sure it's more than that now. And things that I learned in the military that have really pulled through for me, just the sense of doing the right thing, integrity, obviously the leadership, discipline, probably a lot more discipline than I wanted to be at that point in my life, but, kind of challenged me, being adaptable. I didn't end up going into the career field that I wanted. I wanted to be medicine and I ended up flying satellites, but learned how to move through that project management and just kind of strategic thinking. So, as you have kids who are thinking about different things, money may be an issue, or they do want to go into the military, ROTC is not a bad way to go. So, how does this translate? So, as I was wrapping up in the military, I was finishing up my MBA that I got from the University of Colorado, and I was focused on marketing. So at that point, I wasn't really sure what I wanted to do in marketing. I knew that I wanted to focus my career back on the medical side. So started really targeting medical device companies. And for those of you, as you look at marketing, as I get into my, kind of career and the roles I've had, you have upstream marketing, which is really marketers who are looking at, the market as a whole. What does the market need? What are trends? What do consumers want? How do I work with, engineers and developers to create those products? So you're really the business arm to the development early on. And then you've got midstream, which is really your go-to-market planning. Once you've developed the product, how are you going to go to market? What are your distribution channels, your pricing, just all of those and launch plans. And then you've got downstream marketing is once something is in the market, how are you pushing it to customers and how are you help driving sales? And then you have field level. And so this is more of your B2B marketing, but really getting in and working with companies to create programs and campaigns to drive products in through, kind of the B2B. So that's marketing. So as I kind of talk about these different things, just wanted to give you a snapshot of what marketing is. So my career journey, outside of the Air Force, it has been in sales and marketing for my entire career. So when you look at Medtronic, I was very fortunate. Medtronic is one of the biggest global medical device companies in the world. I was working for a division that had about 2,500 employees, 800 million in revenue, so large company. And I started as a clinical specialist. I'll go into more of what these positions are, but work my career up through 10 years to a senior manager of upstream marketing. So really working with engineers on new product development. After 10 years at Medtronic, I wanted to try something new. This was implantable devices for the aorta. Went over to Intuitive Surgical. For those of you who don't know, Intuitive Surgical is the manufacturer of the Da Vinci robot. So a surgical robot, the first one on the market. So a larger company, and I was really, still in that senior manager of capital marketing. So this was downstream marketing, really helping sell the product. And then I went over to Bolton Medical, which was an earlier stage company, 300 employees, as a director of marketing, and then was promoted to VP. CryoLife, a little bit of a larger company based out of here in Atlanta. Actually went through a rebrand that I led. It's called Artivion now, and I was their VP of marketing for a while, and then decided I wanted to go into the startup world. I just had this itch to get with an early stage startup and see if I could really help build and drive that. Fortunately, after 4 years of leading a lot of their commercial operations, the company, kind of stood down and they're in a rebuild phase. So right now I'm just kind of thinking about what I want to do next. So working with a lot of startups here in the Atlanta area on fundraising, on their go-to-market strategies, on how they're building their team. So that's what I'm doing now. So that's been my career progression. So what I want to do is just kind of say, what exactly are these jobs, and how could you speak to them, and what do they mean, and how do they align? So my career path started as a clinical specialist. So what is a clinical specialist? A clinical specialist is part of the sales team, and what they do is they actually go into the OR or cath lab with surgeons, cardiologists, and kind of help advise them as they are implanting or using a product in a procedure. So very hands-on, very technical. You have to not only know your product inside and out, but you're trained to know everything else in the OR that's supporting that. So you're just kind of an extra um, mind and kind of consultant for that physician as he's doing the procedure. In that role, you're doing a lot of education with the doctors and nurses on how to use the device and the other products, ancillary products that you need. So I was a clinical specialist for about 2 years. And the lifestyle there is you support a lot of sales reps, so your territories tend to be a little bit larger. So I did a lot of traveling during that period of my life. But the great thing about being a clinical specialist was there was no homework. So at the end of the day, you weren't, finishing up work, going home, checking emails, having to finish up PowerPoint presentations, doing analysis. You kind of left your work in the OR as you left. So that was a great thing. Key insights here. I'm an exception to the norm. I didn't have a clinical background. Medtronic actually wanted me in marketing, but they said go out into the field and learn the customer first before you come into marketing, because uh, you're going to be a stronger marketer if you understand how products are used in the field. So as you're kind of thinking about, this is a job prospect, for people who are exploring, they usually want a clinical background on this. So nurses or med techs or something like that. Mostly individual contributor roles. From a career path from here, you can go into training and education. You can actually then go and work with clinical trial teams. So new products that are being developed and going through clinical trial, they need people supporting those as well. You can make good money. I'll show you guys kind of some of the, kind of bands of pay in these jobs, kind of toward the end. And then you could be a global clinical specialist. So as companies are looking to roll products out outside of the US, they tend to send US clinical specialists to other countries to help with training and to get them up and running. So this was a great first job. As my second job, I worked alongside sales reps, and I got allured by the money. Sales reps make a lot of money. And I thought, well, I'm in the OR, I'm doing the procedures, I'm upselling, I'm talking to the physician. I want to be a sales rep because I want to make that money. So what does a sales rep do? A sales rep, they're responsible for actually selling the device. So they've got to convince the physician to use the device. Then they have to go into the hospitals and sell to the hospitals. So there's a lot of health economic argumentation, contracting that has to be done, and those types of things. They do a lot of entertaining, relationship building. They do have to cover their cases, and they're held accountable for a quarterly number, which is something I realized that didn't suit me well in my career. Travel kind of depends on your territory. There is a lot of after hour, at least in the world I was in, business planning and prospecting for what you're doing, and you had to entertain. And that was, that took a lot of time away from my personal life. So lifestyle here, sales is a lifestyle. People who go into sales and who do really well and make that money, it is their life. I did sales for a year, decided it wasn't for me, so moved back into marketing at the field level. So for a sales rep can be a full career, and that's when it becomes a lifestyle, and you make money on it because you dedicate your life to that. Um, you can move into management career paths. You can go into management. You can move into marketing after you've been in sales, go and be a chief commercial officer. So sales is a great way, if you're kind of cut from that cloth, to then move a career forward. A lot of medical device companies like sales as people who've come up through the sales organization as CEOs, because CEOs are the biggest champion in an organization for selling that organization. So it does put you on a really nice trajectory. Obviously global opportunities, relationships are currency in sales. Okay, so in my career, figured out sales wasn't for me. I got stressed out by that number and just kind of watching it come in. And it's just, if you hit your number one quarter, next quarter you had an increase on it. So you had to beat that number. So there was never any reprieve for me. And I realized that I like to think longer term. So for me, if you looked at my territory after I left, it was one of the most high producing territories 2 and 3 years later. And the reps that stepped into that territory said it's because I built a foundation, because I built partnerships with hospitals. I set up programs, in the community that supported disease awareness, that supported um, strategic partnerships. So all of that took time to build, but once it built, it became a very sustainable business. So the company actually saw me doing this when I was in sales and asked me to come in house, and it was a new position that they were standing up and be one of their first 3 therapy development specialists. So they said, do what you were doing in sales, but support the sales teams in building their territories. So really, this was a great job because I got to develop disease awareness campaigns. So when you see these different disease awareness campaigns, you're seeing a lot of commercials, but this was done more in the field, or you go to the doctor's office and you see those pamphlets that educate you about diseases. Those were made, by my team in this role for our therapy, and we did that type of stuff. So partnered a lot with clinicians and hospitals on marketing events, and work with sales on account-based marketing. This was a great job. I was in the field, so I was still able to kind of work from home, go out, meet with, uh, marketing teams from clinics and hospitals, create strategic partnerships and do that. Evenings and weekends, if we had events, so there was some of that. And then I wasn't, kind of compensated on revenue, but MBOs, so major business objectives. So I was held accountable to how many programs, how many more marketing type benchmarks. So this was a great entry level into marketing. So that's why I say in key insights, this can be an entry level role, or someone with a marketing degree can kind of go directly into one of these roles. It's an individual contributor role and can be management, career paths, marketing, you can go into sales, you can go into business strategy from there, or even corporate development, which I didn't put, but you can go in there because you're building strategic relationships. Um, so from there, I actually was asked to come in-house and really work with our engineers doing upstream marketing. So once again, strategic marketing, where's the market going? What do our customers want? What are the trends? And I got tapped on the back for this because I had been in the field for about 8 years at this point. I knew the customer, I knew the trends, I knew the competition, I knew how to sell in through programmatic things. So they said, hey, we need to figure out what our future is. So come be a voice inside. So for this one, I did a lot of market research, voice of customer, market modeling, and kind of data analysis as to where trends were going and what it can mean for the company. Work closely with R&D and cross-functionally with clinical and regulatory, because I was in a regulated part of this environment. It was a class 3 device, and then work with physician KOLs on what we were going to do. And so ended up, identifying a few different opportunities for the business, building business plans around that, and then lobbying for funding internally. Lifestyle was great, mostly a 9 to 5 job with some travel and MBOs. So upstream marketing, it's not really an entry level position. They usually want you to have some experience in engineering or marketing to take on one of these roles. Um, it is a more analytical part of marketing, but you really get to speak with the customers, talk with physicians, talk with patients a lot of times as to what do they need and what's going on in their disease journey and how do you work with them. Career path is continuing through marketing and business development. And this was a global position because a lot of companies, you're not just developing products for one region. It's you start in one region and then, you build your channels, you build where you're going to go from there. So then in my career, I I wanted to grow my career, so I had done a lot with Medtronic, a lot with implantable devices, and robotics was sexy. This was back in 2000. 13-ish, 14-ish, robotics were just really making a move into the market. So, I wanted to be part of that. And Intuitive was kind of one of the top companies to work for. So I managed to network myself in there and get a position as a marketing manager downstream. And so downstream means the product is already in the field. And what are you doing to drive adoption? What are you doing to drive usage? And I was specifically working with the team that was selling the $2 million robot. And then there were other teams that were helping physicians use the robot during procedures, but I was helping the team sell this robot to the C-suite. So here it was developing a lot of campaigns and programs to sell the product, working with a lot of agencies, external agencies on these influencers, KOLs, and doing some digital marketing and account-based marketing. So downstream roles are great. I think a lot of people, when they think marketing, think of downstream because it is a lot of your campaigns, a lot of your digital marketing, a lot of your advertising, and those types of things. It was a good job, mostly 9 to 5, a little more travel when you're in a downstream position because you're writing with the fields, you're developing KOLs and champions. So you need to be out there with them to develop those. Um, so this is downstream marketers. It can be an entry-level position. It's mostly an individual contributor role, but you can, kind of lead teams, downstream marketing teams as well. Career path here, training and education, clinical trial support. You can do different things from this as well as into marketing leadership. I saw KOLs, key opinion leaders. So when I tend to talk about KOLs, I think about physicians who are highly respected in the area, CEOs, CFOs of hospital systems that are highly influential, and even sometimes PAs and nurses who drive a lot of that education to patients can be key opinion leaders, meaning that people listen to them. So then with my career, those had all been individual contributing roles for me. And then I had the opportunity to become a marketing director. And I was a marketing director in the US, meaning that just the US geography is what I was responsible for. But I was responsible for every aspect of marketing in this one geography. So in one of the first slides, when, kind of talked about upstream all the way through account based. In all my other roles, I had just been in one of those verticals. So as a marketing director, I was overseeing all of those for the US market. And here it was an aortic stent graft again. And so I was responsible for developing the strategy and launching products into the US market. I, developed a whole team, to kind of fill those individual contributor roles like I had had before. And then we executed, kind of in the field with sales teams and cross-functional teams. So being a regional marketing director, you're working closely with sales and other cross-functional counterparts to really drive the product into the market. Um, it's important. Sometimes people don't think about how important marketing is for a sales organization. Sales is really the tip of the spear, but it's a lot of the marketing support and preparation and planning to deliver sales with the strategy, tools, resources that they need to execute when they're out there in the field selling. Um, so this was a good job, mostly 9 to 5 in the office with travel. Um, you know, uh, yeah, I would say the office when you're in these leadership roles is a lot for alignment, um, and lobbying for resources and bringing alignment. I did bring a lot of work home with me. So kind of, the higher you go in some of these roles, there is that work-life balance that you kind of have to think through. Career or key insights. It's a great career growth position from some of these more junior marketing roles into this. Mostly leadership roles. So marketing directors tend to lead smaller teams than maybe VPs or chief marketing officers. So, you're leading teams anywhere from 5 to maybe 20 people. And then the career path, from a marketing director, you can kind of continue up through marketing or become a CCO, which is chief commercial officer, which means that you're leading sales and marketing. And a lot of companies do like marketing leaders as CEOs as well. It depends on what phase they are in. If they're in a growth phase where they need some strategy, they might lean toward a marketing, kind of career person. If they're just driving hard, selling, trying to get kind of a company off the ground, they might lean more heavily towards sales. So global opportunities there as well. So kind of the final role in my career path was vice president of marketing. So I've been leading global marketing teams for the past 10 years. And so think about everything that I was doing on a regional level in the US, now doing it at a global level. So at a macro level. So making sure that you're developing campaigns and launching products and developing products that have a global need, but obviously tailoring them to the specific geographies. So with this team, you work a lot with the global teams on tailoring their strategies, understanding their customers. Your cross-functional work just becomes exponential. In these roles, for me, I've sat at the leadership table, so been reporting directly to a CEO, and really owning that commercial strategy, and then working, closely with sales to implement. So big job. It's been a lot of fun, mostly office based with more travel, because now you're spending more time not only, in the market that you live in, but in global markets. And, as you move up the food chain in marketing, not only are you working on these MBOs, but also you become accountable for revenue as well. So a lot of your pay and compensation is based on, revenue. And there is a lot of after hours work, and then you find yourself in these leadership positions, having to entertain as well. Um, and from a VP of marketing, I think that's where I am in my career, is deciding what do I want to do next. And that's why I'm taking some time to work with these startups to decide, do I really want to take the reins on a company and be a CEO? And there's some opportunities out there in the startup world for me. Do I want to continue in this VP of marketing career path, because I do love it? And is it okay that I don't take the next step? So I'm sorting through that. I call this my eat, pray, love phase. Or do I want to do something more closely aligned to strategy? So potentially pivoting, um, into consulting or something like that. And then also I've worked so much with hospitals that, do I think I could leverage and do something different in the hospital setting and be a marketer there? So, I think, a VP of marketing gives you a lot of opportunities, just deciding where do you want to go, what do you want to do. Okay, these are a little small, but these are compensation estimates and ranges, because obviously there's a range of experience and companies, smaller companies versus large companies, growth companies versus, kind of non-growth companies, as to what they'll pay. But as a clinical specialist, you're starting out, you might be in that 65,000 a year range, and you can go up into that 150, as a kind of a career clinical specialist. Sales reps, as an entry level sales reps, you're making about 80,000 as you're running. But if you become a career sales reps, 300, you get with the right company, you get with a startup, you get with one of those blockbuster drugs, things like that. I mean, these guys make a lot of money. Anyone else questioning their degrees with the cash flow numbers? I'll take questions in a sec. Therapy development specialist, kind of making similar as clinical specialist. Then you've got your marketing managers, in the low 100s to, kind of mid 165-ish. And then you've got your leadership positions, which you start to make more money. More senior positions, obviously there's more potential. And then there are other junior level positions when you start looking at content creators or, kind of event planners or those types of things, where you can make other money. But, obviously those weren't in my career path. Okay, couple more slides and then I'll take my slides off and I can answer some questions live. So why does this matter to my students? I just tried to put myself like, why are you telling me this, Megan? Why do I care? I don't know. So I thought, why might you care? Why consider a career in life sciences? There's a lot of stability and growth. So even as the economy wavers, I feel like in the life sciences, there's been opportunities. and growth. So I feel like there's been strong job security, and healthcare is rapidly expanding. So that's been a good thing for my career. I'm a curious person by nature and a competitive person. So, innovation and technology. There's a lot of innovation and technology when you're on, the industry side of healthcare, whether it's medical device, med tech, or even pharma. Um, so if you're a curious mind and like being on the cutting edge, there's a lot going on in healthcare there. Um, I would say purpose. I changed this out from something I had in my first presentation. I would say early in my career, purpose wasn't as big of a deal. It is a driver now. The companies that I'm choosing to work with, if I choose to go back into a full-time role, I'm looking at what is their purpose and what is their culture. And I think in the life sciences, healthcare sector, there is that. And the companies that I work with, they don't just preach it, and they're not just sayings that are hanging in hallways. They believe it. And for me, that's been a driving force here this later part of my career. And then I think it's a well-paid industry. So competitive pay, great benefits. And so all in all, that's just kind of supported me and kept me in this career field. Um, so for as you're looking at your younger kids who are still, probably 10 years or so, I don't know, away from having a real job, things that really teed me up younger in my life that I took for granted or I didn't strategically plan for was just sports participation, just discipline, teamwork, and as people are hiring, and even as I hire, kind of people who are coming, into their entry level positions, you love that competitive spirit and that teamwork. So I do look for that. I look for leadership experience, people who have wanted to put themselves out there. Um, and it doesn't mean that they have to take on a leadership role, but I think being in a leadership role um, kind of signifies that you work well with other people, that you're adaptable, that you can influence. And I use the word influence because it's really, can you inspire? Can you, kind of rally people to move in one direction? So it could be indirect or direct leadership experience. I love people who join clubs and get involved. Now I say networking, because that's a big thing as you move your career along. It's networking. Who are you bringing along in your career? So when you think about your students, who are they involved with now? What are they getting involved with? Because that network will support them as they think about where they want to go to college. And then in college, what are they doing? And then education. I don't think every successful career in medical devices has to come through, the 4-year college and in grad school like I did. I think vocational schools work as well. And that's what I just want to stay here, undergraduate degrees. Nursing is a great one, for clinical roles, OR support, clinical trial, business, marketing, corporate strategy, sales. But there's other ones that can work as well. Vocational training, if you have students who don't really want that 4-year commitment or they're not really sure what they want to do, you vocational training for, med techs for that who are running C-arms or, any type of med tech there. It's a vocational school. They can then become clinical specialists and have a really good career and that they can be research monitors for clinical trials. You don't have to have 4-year degrees for those. So it's just kind of something to think about as well. And then I'd say graduate education. I think that becomes more important the further you move your career along for leadership roles. Um, so that's what I wanted to share with you. So I'll stop sharing now and just kind of take some questions if you guys have any. And if you put it in the chat, if you just want to kind of ask me.
I don't have any questions, but I will say, I will agree with the sales reps making a lot of money. I used to be a sales rep for Sealy Mattress Company, and nobody thinks about, selling mattresses, but it was one of the best jobs I ever had.
It's a hard life though. I mean, I have so much respect for sales reps and what they do because that year of my life, there was more rejection and more tears than
Yes,
there's been at any other point in my career. Um, it just was not the cloth that I was cut from, but I love supporting reps. And I think
hmm
carrying the bag, going through that experience, it became to me, it's not all about the money. You got to find a
right.
career path that speaks to you
I
and
agree.
what's going to make you happy. And
I
those dollars didn't make me happy.
Yeah. I think it made me maybe a better teacher because when I got in front of the students, um, I had thick skin. So I was like, there's really nothing you can say to offend me because I've been told no a million different ways.
Yeah. Someone wrote, anyone else questioning their degrees with the cash flow numbers? Can you just explain?
This this is an ongoing like thing that happens with speakers that teachers start questioning their career choices.
I'm
Um,
sorry. I was,
Oh, no, no, no.
it's because I tell my students all the time, I'm like, you're easily gonna go make more money than me. And I mean, you could go do a lab tech job and make almost half my salary and not be in college debt. So I always used to make that joke, like, y'all are gonna go make money than me. So it's a joke, but not really.
Yeah, and I mean, I think it just comes down to what makes you happy. And like I said, even with that sales rep job, and there's been different points in my career where, I'm up and down. I was up and I'm taking this hiatus right now. I call it my eat, pray, love phase because I was like, do I want to continue doing the same thing? Is that challenging? Is that making me happy? And if I go work for a startup, I mean, that's working for the hope of making money at some point in my life. So it's that, but it's, kind of trade-offs. And I don't think that was instilled in me early enough about doing something that makes you happy. I think it was, the influence circle that I grew up in was really a lot about making money. And I question would I have done something maybe a little bit different, if it had been, what is your passion?
so one of the questions was for data analysis as a career, what would be the requirements or coursework for that?
Yeah, so I think for data analysis, a lot of finance type stuff when you get into data, analysis, because market modeling is a lot of what is the return on the investment. And obviously you learn how to run Excel, how to run those types of, kind of other programs. Their statistics is coursework for more of the data analysis type thing. When you get into marketing, there's a lot of opportunity assessments that teach you a lot of the analysis that's specific um, to marketing. And then as I've kind of progressed in my career, just all the stuff with digital marketing. I mean, you've got to know those back ends of those Google Analytics. You've got to understand how your CRM is running, how to download reports, how to kind of clean that data. And a lot of that I've learned just through coursework through the internet, a lot of free courses. I've paid for a few here and there. I invested heavily here this past year into AI, so spent way too much money to take a course that not only taught me how to look for opportunities to integrate AI, but then really rolling my sleeves up and working with the tools for that. So that's going to be important in a marketing career.
And then I think the last question that was from two people is, are you open to speaking with students?
Yeah, yeah, and I'm local. I think I saw that one pop up, and I am local. I live in the Midtown area of Atlanta, but happy to speak with students.
Awesome. And I did put your email in the chat for them.
Okay.
So anybody that's interested can reach out.
Yeah, please do. Please do. I'm passionate about this. I think it's been a great, kind of career journey for me. And I had no idea these roles were out there. So,
Yeah, it's really great that you can point out that other side with the marketing because we talked last week with somebody who's kind of on the back end with developing devices, and that is a back end job, but this is more forward facing. So there are students that fit both of those personalities. So you guys as teachers know who would be good at marketing versus who would be better in a lab all day.
And those guys in a lab all day need a marketing counterpoint, or, because if they're tinkering in a lab, but they don't understand how it's truly going to be used in the field, that's where if you do have someone who's on the cusp of they're not really sure, there's synergies there.
Yeah. Awesome. Well, thank you so
Right.
much, Megan, for
Yeah.
joining us.
Of course. Of course. Yeah, and you guys have my email address, so feel free to ping me if you have additional questions. And other than that, I feel like we're about 10 minutes over time, so I apologize. But good luck with the rest of this coursework.
Yeah, thank you so much.
Yeah, of course. All right. Bye, y'all.
Bye.
Bye.
All the little hearts. Yeah. So glad we got to hear from somebody in marketing. It's a little bit different. At the depot, if you guys go there or if you Zoom with me, I have the biotech careers poster behind me from Biorad, which I will try to get you guys each one for the January training to put in your bag. But basically, it just shows all of those extra support jobs that go into making sure that life science products are getting out there. So with that, it is time to turn it over to Cynthia for our lab.
I apologize that those of y'all who had Trish last week, you're stuck with me today, but my crew was fun. So we'll start off the lesson with tell me your teacher or show me your teacher without telling me your teacher. I came home and I had on overalls today and I just found a pair of scissors in my pocket from an activity I was doing with kids. So yeah, like I was like, oh, that's what that is. Let's get rid of that. So if you don't have your stuff, I have a lovely display over here. You need your 24 well plate, your syringe with your column chromatography cartridge, your C18 pack, graduated cylinder, your 70% isopropyl alcohol, and y'all have beakers, but I have fun little test tubey things, which you can get free from the depot from Megan. You can get a bunch of them. So, oh, and your Kool-Aid packet. And your notebook. Y'all don't have page numbers, but it says Kool-Aid chromatography up the top, and your safety glasses and your gloves. And I'm really jealous of the woman who's sitting at the beach with her daughter who's in a swimsuit. That's just rude. Rude.
Yeah, Pam's on fall break.
Sorry, it's
This
not
is
rude,
Pam.
Pam. It's quite nice. You should let your kid do this lab. She'll love it. How old is she?
How old are you?
Oh, see, I saw that face. You better get her stuff and let that sweet cherub do this lab.
I've had her sitting here practice
Okay,
micropipetting.
cool
Right?
girl, you can do this. This is totally easy. There is a little bit of math involved.
Are you good
So this is column
everybody?
chromatography, and you can do this with the C18 cartridges. I will tell you this, these suckers are really expensive. You can't buy these on Amazon. You have to buy them from a very specific company. I bought like 11 and spent a lot of my own money so I could have these, but these is expensive. You
So
can do this with
I'll
paper.
I'll interrupt you for a second,
Go right
Cynthia.
ahead.
So fun facts. Some of you have this in your box. That's a card from Sorbtech.
Yep.
But if you flip through your binder, there is a column care document. And so on there, it's got an email for a guy at Sorbtech. They sell a box of 50 of them for about $150. So they work out to $3 a piece instead of the 11 that Flynn charges.
Yep, I got mine through Flynn. So yeah,
Yeah,
thanks for letting me
yeah,
know that. That was years ago though.
yeah.
But as long
Um,
as you take good care of them, they will last. They will last a really long time.
Yep. And so if you request that for implementation, we'll send you a set that then you can just rinse out and keep using. So, all right, I'll turn it back to you.
No worries. And if I don't do a good enough job of explaining this, there is a YouTube video. It's called Steambox, and they do the exact same thing. It's this cute little girl, and she explains everything, except she doesn't do the math. She kind of wings it, but the math is really important. So the main math equation that we deal with is your concentration and volume. It's in your notebook. It's C1 V1 times or is equal to C2 V2. The first part of your instructions, if you have not done it already, is that you need to make your Kool-Aid solution. In your instructions, it says like 0.3 grams. You can use a food scale, or if you have a major scale, or you can kind of eyeball it. It's just not the whole package. And then you're going to fill up your beaker with 50 milliliters of distilled H2O. So if you have not made your Kool-Aid, please make your Kool-Aid now.
Question.
Yes.
So the directions just said to put some Kool-Aid in some water. So that's where we went. Is that going to be a problem? Do I need to
No,
be
it's
more
not going to be a
precise?
problem. It's
Okay.
really not. I mean, I've done the whole package before.
Yeah, I just kind of made a
You
a
good
bottle.
girl. If you did the
Okay.
whole bottle, that's totally fine because you can save
Okay.
that and use it for your like your whole classroom.
Got it. Perfect. Thank you.
Yeah, no worries. I eyeball it myself as well. But if you want to be precise and if you want your kids to be precise, it's 0.3 grams of Kool-Aid to 100 mils of water. So if you're teaching like Uber techniques, those are the precise measurements. And I have a lab that this is written up that's like super kid friendly, makes them work out the math problems while they do it as well. It's like this, but like a little different. So you're going to do math. So if you are not good with math in your head, you're going to need a calculator. You can use your cell phones if you like. But you're going to take one of your beakers and put in 70% isopropyl alcohol. I've got one that's just got 50 mils in it, or you can pull directly from your container. And we have to make 2 more solutions. Depending upon if you're a block schedule class or if you are a like 45 to 50-minute class, you might want to do prep work the day before. It also gives kids an opportunity to like do a web quest on chromatography and the ways that industry can use it. And then on that day, they can do their math and create their solutions. And then the next day, they can do the chromatography part. Just depends upon how advanced your kids are. So there's different ways to implement this.
I'm sorry.
Oh,
How
I didn't
much
hear that. That was
again
good.
of the 70% into the beaker?
So I put 50 mils in because we're going to use the 70% to charge our C18 cartridges, but we're going to use these 50 mils to create our 25% solution and our 5% solution using the C1V1 equals C2V2. And we're going to walk through the math.
So I think we only have 2 beakers. Are we able to use the little cups for the smaller concentrations later?
Yeah, we can adjust the volume for that. That's not a problem. Let me see how tiny your solution is. Sorry, I'm trying to get a box. Well, where'd they go? There they are. Okay, what are these?
It should hold 10 milliliters.
We
So
can adjust for 10 mils because that's really all you need. We can adjust the math. We'll do extra math. Okay, so I'm gonna do math. All right. Megan says y'all can see this correctly, so I'm just gonna write it. So your equation is C1 V1 equals C2 V2. Does everybody see that? Okay, cool. So your C1 counts for the concentration of your original alcohol. Let's just say that your school has 95% alcohol. You can do this with 95% alcohol. Then you're just going to make a 70, a 25, and a 5. You can do this with any percent alcohol you have. You just get to do different math. So if you have 70% of your original concentration, We are trying to figure out the volume of how much we need. So this is our variable. If we want to make a 25% solution, that's the concentration C2. So that's 25 here. If you're going to use the smaller little cup, make it 10. I have a larger container, and if you have the beaker or a container in your school, 50 mils is roughly all you're going to need. So I'm going to stick with my original, that my volume that I'm going to create is 50 mils. Is everybody with me so far? Okay, Vicky, that made a face, so we're going to go back over it. Yes, I'm watching you with your cameras. So your original concentration of the alcohol that you're using is 70% isopropyl. That's your C1. Our variable is how much of that 70% isopropyl alcohol do we need to create the 25% solution. So 25% is the concentration that I want to make. The 50 milliliters is what I'm going to fill my container up to when I add together how much alcohol I need plus how much distilled water I need.
Okay.
So if we do some basic algebra, we get 70x is equal to, and I did this earlier, but you can do it with your phones, is equal to 1,250. And since we want to solve for x, we're going to divide by 70. And I'm going to pull over my nice little handwriting now. Ooh, I can't see my own screen. Where am I? Screen. Oh, there I am. So if you do the math, you're going to end up with x equals 17.8 milliliters. That 17.8 milliliters, it's hard to do on a graduated cylinder, so you can actually increase it to 18 milliliters. So you're going to put 18 milliliters of isopropyl alcohol into your 50 ml container. And since we're going to a volume of 50 milliliters, 50 minus 18 means that the remaining 32 milliliters is going to be your distilled H2O.
Or tap water if you don't have distilled.
Yeah,
water.
if you want a
Maybe
pinch,
okay.
you can use tap water. I wrote DH2O because it just teaches the kids, like, there's distilled water, there's double distilled, so like DDH2O. Like, you can get all creative on the different types of waters that kids can use. So using your graduated cylinder, which you've been provided with a 100 and a 10, you're going to measure out 18 mils of IPA, which is your isopropyl alcohol, and 32 mils of water. If you went with the smaller cup, I will do the math. So if you have 70%, you're making a 25%, and you only have a volume of 10 mils, that's 250, 70x divided by 70. X equals,
I have a question. Are we supposed to have the little containers with the blue caps?
no, those are extra. That was in my prior statement that I
Oh,
got
okay.
those from Megan's Depot,
Okay,
but you can use the beakers that she's given you.
just making sure.
No worries. But I was trying to plug the depot. So if you're using the smaller 10 mil cups, X is going to equal 3.57. So you would put
You slide
4
it up.
mils of 70% isopropyl. And then if you're using 10 mils, that means you do 6 mils of distilled H2O.
Slide it up, Cynthia.
Oops, sorry, my bad. So yeah, if you're using the tiny cup, it's 70 times X is equal to 25% with 10 mils of total volume. And when you do your math, your X, which is your amount of isopropyl alcohol, would be 3.57. You can round to 4 mils. It's a little bit easier. So it'd be 4 mils of your 70% isopropyl, 6 mils of your distilled H2O. If you're using the smaller cups. I have a bigger cup. So I'm going to do my thing, and then we're going to do the 5% solution. Oh man, that was a perfect pour. The fun thing about these little vials that I got from Megan, they actually have a graduated cylinder along the side. So I can hit it to 18 and then just fill the rest of the way up with distilled water. It's a built-in graduated cylinder. Once you add your water to your isopropyl alcohol, give it a couple of inverts, give it a good mixy. All right. I'm going to wait until I see that we're ready to move on to solution number 2. Uh-oh. C is concentration. Sorry, thank you, Kennedy. I said it out loud, but I guess I should have written it. My apologies. Okay. Are we ready to move on to our 5% solution? Give me a thumbs up if we're ready. Lovely. Do I have students that need a little bit more time? Say yes.
Can I
Okay.
see the first? I'm sorry.
Yes, yes,
Can I
ma'am.
see the first? Yeah,
I got you.
because I have the little tubes.
Okay, so if you've got the little tubes, you're going to do 70%.
Mhm.
Your X is your variable of how much of that 70% we need. And we're creating a 25% solution with that 70% isopropyl. And since I have the little tube, it goes up to a volume of 50. I like to work in perfect numbers. So if you divide, you're going to end up with 17.8. So I just rounded and did 18 mils of IPA. And since my total volume is 50, that means I would fill the remaining 32 mils with distilled water.
Okay.
No worries. Okay. Let's move on to our 5% solution. So we're still using this C1V1 concentration. So we're still using 70% isopropyl alcohol. I still don't know how much I need of it, so that's my unknown volume amount. But now I want to create a 5% solution. I'm still using 50 mils. If you're doing 10, we'll make another equation page for you in just a second. So if I get my variable by itself, I will end up with 3.57 mils or 4 mils of IPA. And then the remaining 46 milliliters will be of distilled water. And again, depending upon how math fluent your children are, it could be that you need to make solutions the day before and make them get all of their equipment and their inventory together and walk through the procedures with them before you get into the actual doing of the lab. And I know that this lab page had extra questions for the kids to work on to get more proficient with C1V1. So if you were using like the 50 ml beaker, you can go ahead and rock yours with the 4 mils of IPA and 46 milliliters of distilled H2O. If you're using the tiny cup, let's do yours. If you are using the tiny cup, we're still using the C1V1 equals C2V2. So we're still using our 70% isopropyl alcohol. I don't know how much I need of it. There's my variable. My concentration, I want to make a 5% solution. And if you're using the tiny cups, your total volume is going to be 10 milliliters. So that's going to be 70x is equal to 50. Ooh, this is not going to divide well. This is going to be minuscule. So if you have at least one of the beakers that Megan has for you, we might not want to go this tiny. But if we do divided by 70, ooh, that's going to be like 0.7. So you could round it to 1 mil of IPA to 9 mils of your distilled H2O. That one's going to be a stretch, ladies and gentlemen. But that is how you do the math.
Does this math work with any
Yes, ma'am,
solution,
it does.
even if it's not percentage?
So all of the alcohols that you buy are going to say like 70, 90, 95. So you can easily manipulate any of the percentages of the IPA that you can buy from a scientific catalog or what your school might have on hand. Even at the grocery store, sometimes you can find like a 90% isopropyl. You can still take the exact same equation because you don't need 70. That's just what Megan gave you. Like if you went to the grocery and you found a 90%, you're going to put in 90 for that original concentration of C1, and then you're still going to solve for X, and it would still be a 5% or a 25%.
And this would work with like other alcohols as well, or even like hydrogen peroxide.
You'd have to check. Okay, Megan's nodding yes. I was about to say, I was like, I don't know straight offhand, but I'm sure that you could work with the C18 cartridge and see what it would do from a column chromatography standpoint. But Megan is nodding her head yes.
Yeah, and then any like stock solution that you're using is going to say how concentrated it is. So like with the buffer, we're going to be using a 50x. So you would plug that in as the concentration.
Got it. Okay, so I could use it for other
Yeah,
situations.
and
So-
molarity works the same.
So if it's like 3 molar.
Yeah, 3 molar,
you
and you
could
want a
use the
0.3 molar. Yeah.
Yay, great. Okay, I could use this for good research purposes. Great. Awesome. Thank you.
With the hydrogen peroxide though, would it interact negatively with Kool-Aid?
I don't think it would work great with the Kool-Aid, but if you were doing it for something else, I think that's why she was asking.
Oh,
Yeah.
okay, cool. I was just double checking because I was like,
So,
I don't know
like,
if the hydrogen peroxide would
we
react well
did
with Kool-Aid.
we did like the enzyme assay like with potato extract, which that smell will like haunt me forever with the ugh. But yeah, so diluting the hydrogen peroxide, use that exact same formula to make the dilutions.
Got it. Okay. I'm a research teacher, and sometimes having them, it's been a while since I've done this math, this thinking. So that should be good. Thank you.
Yeah, good to bring it back to the top.
This is a very, for those of you who teach biotech or want to teach biotech in the future, C1V1 concentrations is a very, like, they got to know that one for their pathway tests. And M1V1, they got to know that. It's got to be memorized. Okay, sorry. I got to make my solution. All right, so everyone should have their Kool-Aid and their 25% and their 5% solutions. If you don't, wait until everybody's there. We don't really need to move on if you don't have that done. And again, once you get them in there, give them a little mixy. Pam, how's your daughter doing? Or did she bail?
She told me this is not the fun part yet. Call her back when the fun part comes.
Well, it's all fun. Okay. Somebody give me a thumbs up.
She really just wanted to play with the Kool-Aid, and I had already made the Kool-Aid, and she was like, "When do we get to play with the Kool-Aid colors?"
Well, you can yell at her. It's next. All right, give me a thumbs up if we're good on solutions, that you have got your 5% and your 25%. From my group from last week, have any of y'all started your book read? Those of you who had never read Gila before? Mindy, I saw that.
No, I haven't. Sorry.
Girl, I'll mail it to you.
Okay.
I got
checked
extra copies.
I checked my library. We have a class set, so I'll be incorporating it. Yes.
Girl, I will send you my project stuff. Send me your
Please
email
do.
address.
Okay, I'll put it in
And
the
I'll
chat.
send you my breakout box. Life changing.
And I'm
Okay,
still waiting on my
share.
book. My library doesn't have it. So
Shame on your media specialist.
I would like the project though, but I'm waiting on the book to come. We're getting it from another school.
Good, good. Everybody needs a class set. Oh, sorry. The book is The Immortal Life of Henrietta Lacks. My group, we had a book discussion. It's an amazing book. It's life changing. You should read it, especially if you teach biology. You could do a book discovery, which would then hit ELA standards in your classroom. It's a great book. Not put it down. It's only like 200 pages too. It's not long. Okay, I digress. Let's get into our Kool-Aid. So the first thing that you have to do is you have to charge the filter. Here's the big thing about these filters. You never suck anything up the filter. Things can only go down the filter. So if your filter is connected to your syringe, please remove it. You can set it in one of the last sections of your well plate. Okay. So the first thing that you're going to do is you're going to take 10 mils and you stick with 10 mils the whole time. You're going to take 10 mils of 70% isopropyl alcohol and put it in your syringe. So everybody start there.
Do you take 10 mils even if you only made a total of 10 mils?
Okay, that's your solutions for later on. This is
Oh.
70%. Sorry, we are only pulling from 70.
All right, I'm sorry. Can you tell me
No,
again?
you're fine. So you're going to pull up 10 mils of 70% isopropyl. It's the stuff out of the bottle.
Oh, yes, I see. Okay.
Yeah, those solutions are for later. Don't worry. Okay. Spam. I'm gonna let something go away. Okay, so if you look at your cartridge has like a wider opening and a smaller opening. The wider opening is what goes on the bottom of your syringe, and you should be able to read the word C18. So put your syringe connected to your cartridge. And this is called charging the column. So you're going to push your 70% isopropyl alcohol through your cartridge. This is going to take more than one of these wells. If your school happens to have a 6-well plate, this does work well on a 6-well plate, but if you have a 24, it works. Or if you have a bunch of these tiny little medicine cups, you can use the medicine cups with your kids as well. So you're going to push the 10 mils of 70% isopropyl alcohol through your syringe.
Okay, I'm trying to figure out, y'all, I'm sorry.
You're fine.
I,
What you need?
oh wait. Okay, Cynthia, do you have two cameras going?
I do. I have my face camera and
Okay,
then I
that
have
one's
my
okay.
lab camera.
Just kidding. Don't, never mind. It's after 5
What
o'clock.
you need?
Oh, because I guess I thought it went still, and I was like, wait, I was like, I want
It's
that to
me.
be the big picture, and then it went to like a participant in the big picture, and I was like, no. Okay, I got
Sorry, my bad.
it. No, I don't, I'm not really familiar about how the camera views work. No, we're good. Thank you.
No worries.
I have her doing a split screen. Like if you ever see anybody like filming themselves while they're doing a video game, it's the same software. So, um, and Cynthia, could you put a white sheet of paper behind it?
Yes, I'm sorry.
How much are you putting in each well?
They fill with like 2 mils, just till it fills up and then go to the next one.
So I roughly filled up about 3 containers, but I went kind of all the way up to the top. So I filled in my alcohol in this first one, second, and third one. So like A1, 2, 3. If you need more, then by all means, but I kind of filled mine up to the top.
So it is okay if we did like 5 of them all
Yeah, that's fine.
across column A?
Yeah.
Okay.
Yeah, you're fine. So now you're going to take your cartridge off of your syringe and you can just set it in the last little well. Now we're going to get 10 mils of your Kool-Aid. And again, we never pull up through the cartridge. So that's why the cartridge comes off every single time. So you're going to pull up 10 mils of Kool-Aid. Once you get your Kool-Aid in, you're going to add your cartridge back to your syringe. And here's where the fun part begins. So in column chromatography, this column, because we have charged it with a 70% isopropyl alcohol, it's going to collect the food dyes, the FD&C dyes. And the first time what's going to come out is the smell. So you can teach your kids a wafting activity. So you're going to take your syringe and you're going to push the Kool-Aid all the way through until it's completely out of your syringe. So we go back to our well plate and push it out. If a little bit of color comes out, don't freak out. My Kool-Aid's a little more concentrated. But you should just be getting out clear liquid right now. And that the FD&C dyes are being trapped in the column. Getting a little too much now. You should be able to see it in your column. See your colors getting trapped? This would also be fun for your kids to take pictures with their cell phones, and you could use this in documentation in their lab notebooks. If you use an online learning platform, the kids can upload their pictures as part of their results and their data. And when you're done, you can actually kind of bring it over to your nose and give it a little waft. You should smell grape.
Or lime if you guys chose the green.
Oh yes, if you chose lime, you're gonna get lime out. Sorry, I went for grape. That's my favorite color is purple. Once you have pushed your liquid, and honestly with your kids, you can take off the label that says chromatography. That way they can see it better. But you're gonna take your cartridge off again. Stick it in your well plate. And now we're gonna pull up 5%. So please make sure that you're going into your 5% solution. You're going to pull up 10 mils of your 5% into your syringe. Okay, so you should have 10 mils of 5% alcohol in your syringe. You're going to put your cartridge back onto your syringe. And then you're going to push the solution through. Now, if you make your kids do like a web quest beforehand, which I normally do, you can have your kids look up the molecular structures for the FD&C dyes, the red, blue, green, orange, yellow, red, and you can make them make hypothesis about which color they think is going to come through next based on size of molecule. You can also make your kids research polarity of molecules because that's what's happening with the column chromatography, and have them make either hypothesis or, you know, a line later about what's the most polar solution versus the most non-polar solution in comparison to the 70, the 5%, the 25%, and the Kool-Aid molecules. It's another fun chemistry thing you can do with it. But you're going to push the 5% solution through your column. Oh, and the magic is happening. How's the small child, Pam? Is she excited?
Oh yeah, it's coming out red.
Ah, so that should tell us something about the red food dye molecule.
And then it came purple.
Yep, they're depending upon like how well you make your solution, sometimes it will get a little bit darker. That's not to fear.
And then blue.
Girl, don't go yet. Oh, you got blue out? Uh-oh. You might have had too much in your solution, but that's okay. It's okay.
I've collected other children too, besides my own.
More friends at the party. I love it.
And I wish we had the microphone on because they were like, "What? This is so cool."
You can totally leave a microphone on for small children. Does she have a question? Am unmute that
Yeah,
microphone.
what's your
I have a
question?
question. I have a question.
Yes,
How
ma'am.
do they change colors? Yeah, how do they change colors?
So, what grade are you in?
I'm in 4th.
Okay, so when you were little, did you ever mix paints together? Did you learn about your primary colors of pigment?
Yes.
Okay, so Kool-Aid, what color Kool-Aid did mom pick?
What color Kool-Aid did we do?
Purple. Purple.
Okay, two colors make purple, young lady.
What's your colors? Look.
Blue and red.
Very good. So what color came out of the solution?
Purple. No,
And
blue
what other
and
colors?
Red. Blue and red.
Very good.
So they took
So
like
then based on that, what color should come out next?
purple.
No. Go back to your
If
colors.
red came out first, what came out next?
Blue.
Blue come
I don't
out?
know. Ours? Yeah, what did we do? So ours came out red and then purple and then blue.
Look, it's stuck.
Okay, you just got a little extra solution. More blue will come out later. If you look
Yes,
at
look over
your
there. More blue's coming
column,
out. You see Sammy?
you should have a blue cartridge right now because your red should have all completely come out.
Oh, so maybe we
Last
need
one
to,
was a little blue. It's
because there's
okay.
more blue in here. I see. Yes.
Yes. So you're gonna take your cartridge off. Oh my God, she's learning science. Okay, grab your 25% alcohol solution. You're gonna pull up 10 mils of your 25%. You're gonna put your cartridge back on. And then you're gonna push the 25% solution through your cartridge. And you should see blue coming out.
Yes. Blue is coming out.
Okay, now I still have a hint of blue in my column. So if this happens with your kids, you just need to take a little bit more of your 25% solution and push it through again. So you'll get even more blue because eventually you want your column to be clear. If your column doesn't come clear, don't worry. I'm going to tell you how we flush it to get it clear when you're done. But you would take a little bit more of your 25% solution, and I'll explain to you why this happens. But take your cartridge off, fill your syringe up with 25%, put your cartridge back on, and then push the rest of the blue out. Yes, ma'am. Most of mine came out. You might have a tiny little hint of blue. Don't freak out. I'll tell you how to clean your cartridges when we're done. Sometimes Kool-Aid sneaks in one of those other color blue dyes, and they don't always have to write that on their ingredient list because there's a lighter color dye blue, and you just need a different percent alcohol to push the rest of that out, and that's not a big deal. What we're going to do to make your cartridges all nice and pretty and clear again is you're going to fill up your syringe with 70% isopropyl alcohol, flush it about 2 times. Takes about a day or two, and then your cartridge will be nice and dry and ready to be used over and over and over again. And you can do this with different colored Kool-Aids,
Cool.
but sometimes it helps to look and see what the molecular structure is of the FD&C molecule, because if they're really close to each other, it doesn't separate as well. That's why I feel like the purple one's the best, because of the molecular difference in the red dye and the blue dye.
So we can use these, I mean, reuse these
Indefinitely.
how many times? Like to
Indefinitely.
infinity?
Yep.
Whoa.
As long as you take care of them and you never suck solution up the column,
Okay.
always push down.
Okay. Wow.
I've had mine for years.
Wow.
We just keep sending you a new one every year. So
Oh, I have
adding to the
quite a
collection.
plethora of them now. But yeah, the way that you just completely like flush your cartridge out, take your cartridge off, fill your syringe up with 70% isopropyl, and flush it at least 1 or 2 times, and then just let them air dry on a paper towel. And then the next day they'll be nice and dry and ready to go again. And I just store mine in a sandwich baggie once they're dry. Another thing you can do is if you fill your syringe up with air, if you're like really nervous about it, you can push air through the syringe. Like instead of like right now, I'm doing a flush with 70%. And then because I like to make sure that there's like no liquid in there, because I'm always nervous about it, is I'll take my syringe off, or take my cartridge off and fill my syringe up with air and then push air through.
Oh, that makes sense.
Yeah, it helps to get out a little bit more. I do that a couple of times. It helps to get more of the alcohol out of the cartridge so it's a little bit more dry. Otherwise, it'll take a little bit longer. So yeah,
So
if you
if
just
a,
push air through it a couple of times after you've done at least one flush, it'll help to get it a little bit drier, faster.
if a student does suck up liquid into it because we work with children, is it like
Yeah,
ruined?
it will.
Yeah. Okay.
Yeah.
Good to know. So
No,
I can make it
sorry.
very clear.
Well, depending upon how your kids are, you may have to put the important kid in charge of the cartridge.
Mm.
It might be
Got
that you're responsible for making solutions, but you're responsible for my cartridge.
Gotcha.
It's like that kid doesn't get to run with scissors. Yeah. So there you go. And that's how you do it.
Or even take the scissors home and have them in their pocket when they
Yeah,
get
you know,
home.
I'm that kid, but my kid, okay, so I teach earth science. So my kids made a scale model of the earth. Every kid did a strip, and then we put all of the strips together. And I have an entire earth outside of my door, so I was cutting tape all day, and this was a really nice pocket to keep my tape handy. It looks epic. All the kids were like, "That's so cool," and I'm like, "I know, you made it. It's beautiful."
And where did you say we could buy these? The cartridges?
So if you flip like 1 or 2 pages in the chromatography, the last page has care instructions.
Okay.
And then at the bottom, so Trey Massengill works for Sorbtech, and
Okay,
he's
got
the
it.
sales rep that I work
That was
with.
the card you were talking about. All right, it makes
Yep.
sense now. I'm
And
on the
so if
delay.
you just
It's
say like,
so
"Hey, I was in the training," there's the
perfect.
card too. Um, they will they'll help you get it.
Thank you.
You're welcome. And
But again,
I think ship for free. Like they don't charge for extra ship. They're just like, "Do you want to ship it?" And I'm like, "Well, I'm down the street. I'll pick it up." So save your money. But yeah, so I think shipping is included in their cost.
Amazing.
and again, like if you're doing lab protocols with kids. They can take pictures of their well plates when they're done. They can take progress pictures of their cartridges while they go along, and document those in their lab notebooks.
Speaking of that, do you need us to take a picture for a deliverable, or are we
Yes,
good to kind of
please. Yeah,
clean?
take a
Okay,
picture
perfect.
before you pour it out.
All right.
And then clean up, you can just pour it out over a sink and rinse it out.
Oh, I'm gonna copy the YouTube video that I was talking about. I mean, and guess she does a good job. She doesn't go over the math, but I am gonna copy it and I'm gonna put it in the Zoom chat in case you need it. Or like if you have a kid that's out, they could watch that as a tutorial prior to your lab day. I oftentimes put a lot of YouTube videos on my class site because a kid might need a refresher before they start doing the lab. And if they want to take that time and dive into that video to watch it so that they don't screw up, then by all means, let them watch video. Ooh, so how is this used in the biomedical environment? Funny you should ask. Oh, yeah, I see the HPLC systems. Yep. And gas chromatography. So in forensic science, if we find solutions and we're not really sure what they are, we can do chromatography because all items separate at different solutions. If I know that the FD&C market does this a lot to determine the concentrations of the solutions that they have, things like in shampoos and conditioners, your detergents. I'm trying to think of all the other things. I have a whole web quest on chromatography where the kids learn about different things in industry that use chromatography, paper, column, HPLC, gas chromatography.
Well, and within column, you can do different agents in there. So they can do it by affinity, like there are the affinity columns, and then you can like change the pH so it'll drop different products at different pHs. And
Yes, Carla.
yeah, there are all kinds of different things you can put in
Yes.
column.
Pretty much every industry, environmental, agriculture, they all use some form of chromatography.
Hey, y'all, sorry to interrupt. I've got senior night for volleyball starting in 3 minutes. Am I okay to go ahead and cut out?
Bye, Andrew. Have fun at senior night.
Thanks, y'all.
Yeah, thank you.
I mean, technically, if you think about gel electrophoresis, it is a form of kind of a chromatography of separating out bands. I mean, you can kind of make a lot of connections with it. I mean, I know it's using an electrode and fragments, but it is a separation. Does anybody have any other questions? God bless you. I didn't hear you, but I saw you. I told y'all I'm watching.
All right. And I will stop recording. Yes.